Hello friends, welcome you in my YouTube channel. I am Dr. Mohammad Sirajul Faizi, Assistant Professor, Department of Chemistry, LS College, Muzaffarpur. Today in this video, I am going to discuss about proteins and protein functions. So look at this picture. These are food which rich of proteins. So what is the basic unit of proteins? So first look at the hierarchical organization of proteins. Backbone, side chain, this is amino acid and primary sequence. And now you have the peptide, you have residue and peptide bonds. So if you protein GB1, 3GB1, you see alpha helix and beta seated. Myoglobin, here also alpha helix. So natural amino acid, nearly all polypeptides from animals and plants are constructed from the 20 standard alpha amino acid. All alpha amino acid in L configuration except glycine. Side chain vary, essentially 10 versus non-essentially 1. Coexist in two forms, ionic, zwitterionic form and unionized. Look at here. This is the carbon. This is the alkyl group variant. This carboxyl COO minus and NH3 plus amino and hydrogens. This will vary, so it is called variant. So what is alpha amino acid? Alpha amino acid is an organic compound that contains both an alpha amino group and a carboxyl group bound to the same carbon. This is the carbon, this is the COOH groups, this is the NH2 group, alpha amino group bound to the alpha carbon, so alpha amino acid. Classification of alpha amino acid. Classification of standard amino acid, nonpolar and polar. Polar further classified into three: neutral, acidic and basic. Nonpolar alpha amino acid, glycine. A nonpolar amino acid is an amino acid that contains one amino group, one carboxyl group and nonpolar hydrophobic side chain. Polar neutral alpha amino acid. A polar neutral amino acid contains a side chain that is polar but neutral at the physiological pH. Side chain can form hydrogen bonds. So look at this structure of polar neutral amino acid. Polar acidic alpha amino acid. The polar acidic amino acid is an amino acid that contains one amino group at two carboxyl group, the second carboxyl group being part of the side chain. Look at these two pictures. This compound contains two COOH groups. This is also two COOH groups. Polar basic alpha amino acid. A polar basic amino acid is an amino acid that contains a polar basic amino acid is an amino acid that contains greater than two amino group and one carboxyl group. The second amino group being part of the side chain. Look at here. It contains greater NH, here NH2, here NH2. So these are polar basic alpha amino acid. 3 letter and single letter codes. Amino acid 3 letter and 1 letter. alanine, ALA, one letter A, and like so on. Chirality alpha amino acid. Alpha carbon is a tetrahedral stereocenter except glycine, pair of enantiomers. Only alpha amino acid, L-alpha amino acid, exists in the protein of animals and plant, with very few exceptions. Amino acid refer to L-alpha enantiomers, D-alanine, L-alanine, D-glyceraldehyde, and L-glyceraldehyde. So this is the glycine, COOH groups, NH2, and these two hydrogens. Acid-base properties. COOH group will convert COO minus H plus. NH2 take this proton, become NH3. So low pH, neutral, and high pH. So isoelectric point, deuterion. So look at here, mostly deuterion in this case. Electrophoresis. Analyze a mixture of alpha amino acid, identify substance in an electrical field by separation. Cation move to the negative electrode, anion move to the positive electrode. Neutral alpha amino acid does not migrate. List positively charged, glue negatively charged, Phi neutral. So alpha amino acid is the list. This is the list of alpha amino acid. Basic chemical reactions. Backbone common to all amino acid. Amines and carboxylic acid undergo dehydration to form amides. Peptides are the polyamides formed by alpha amino acid. You see, this is dehydration takes place here, and peptide group bond. Side chain amino acid specific often occurred as post-translation modification, signaling natural modification, oxidation, damage. Disulfide bond formation between cysteine and important structure feature. This is disulfide bonds. If you have the compound where is the SH, then so oxidations, sulfide bond. After reductions again. So a small protein insulin has two polypeptide chain connected by two interchain disulfide bond. There is also one interchain disulfide. The amino acid cysteine contains a thiol group SH. Pairs of cysteine residue often link two peptide chain or two parts of peptide through the disulfide bridge. Formation of disulfide is an oxidation reaction, and the reverse involves disulfide reduction. So summary, protein overview, amino acid chemical composition, backbone and side chain, classification, side chain properties, physical and chemical properties, deuterion form, peptide bond formation, peptides and protein, polyamides, heteropolymers of amino acid. So friends, hope this video will be helpful to you. If you like this video,